Thank you so much. I'm excited to be here. Again, my name is Ivana Parker. I'm an assistant professor at the University of Florida in the Department of Biomedical Engineering, and excited to share my work with you today. So in the Parker Lab, we care a lot about chronic inflammation, and we care a lot about chronic inflammation and specifically how that increases risk for HIV transmission among women. So one of the major issues that we look at, or diseases that we look at, is bacterial vaginosis. And one of our goals is to improve therapeutic targets for bacterial vaginosis. And we do this using a mix of omics, proteomics, bacterial genomics. We do a lot of metagenomic analysis of vaginal fluid, bioinformatics, and systems biology. And so why do we care about BV? So bacterial vaginosis lacks effective therapeutic strategies. It's the most common vaginal syndrome among women of reproductive age. So 1 in 4 women in their lifetime will get bacterial vaginosis. That affects 1 billion women globally. It frequently recurs. So even if there is a treatment, generally it will recur 50% of the time. And the main strategy for treatment right now is antibiotics. So one-size-fits-all approach is antibiotic. Other adverse effects that are related to bacterial vaginosis include increased HIV risk, which is how I started to be interested in the disease at first, but it's also linked to increased risk for preterm birth, cervical cancer, and pelvic inflammatory disease. So what is BV? It's a shift from a lactobacilli-dominant microbiome to an anaerobic dominant microbiome. And it can be symptomatic or asymptomatic. The symptoms, if you do have them, include itching, burning, extreme discomfort, and you can have this dysbiosis with no symptoms as well, which also occurs in many women. So the way that it's diagnosed is via Amsel's criteria, where you'll have vaginal discharge, clue cells, which are cells that basically are epithelial cells that you can get from a swab that have bacteria kind of all around the outside, a whiff test, or a pH of greater than 4.5. And this is a syndromic diagnosis. It's also diagnosed via Nugent scoring. So Nugent scoring looks at the types of bacteria via a gram stain. So you have rod-shaped bacteria that actually are indicative of a lactobacillus morph types. You have Gardnerella morph types, which are more circular. You can see those here. And then you also have Mopiluncus morph types, which are kind of these curved ones. And so there's like a ratio or counting that happens that allows you to be able to diagnose BV based on the ratio of these bacteria that are available or that are seen on the slide. And you can see here, this is a vaginal, normal vaginal flora, intermediate, and then this one would be BV. And so how can AI help? Right? So the microbiome is very diverse. It's also been shown to vary a lot by race and ethnicity, by environmental factors, diet, and things like that. Right? So we would like to develop population-specific therapeutic strategies, or something more along the lines of personalized medicine. That's not just this antibiotic, one-size-fits-all approach that kind of wipes everything out. So this would include altering the vaginal microbiome in an informed manner and using AI tools for hypothesis generation. So what we've really been doing is trying to see if we can predict bacterial vaginosis diagnosis using these microbes that we get from the metagenomic or 16S sequencing analysis. And we hope that this will allow us to find understudied targets, understudied bacteria that are important to study, which we also take into the wet lab and do some in vitro experiments, but also that allow us to understand why certain treatments may not work across the board. So high throughput sequencing has allowed us to really determine the microbial makeup in health and disease. We had a whole session on Monday on microbiomes and AI. So it's really allowed us to understand what microbes are actually composing the vaginal microbiome and get more to that kind of population-specific investigation. So yes. Okay, so the research questions. So we previously, and I'll go through this, had looked at microbial communities in women that were symptomatic and then women that were asymptomatic to see how we were able to predict bacterial vaginosis. diagnosis in those cohorts. Here, I shared that bacterial vaginosis is also increased risk factor for HIV transmission, but we wanted to actually understand, are the microbiomes of women who are living with HIV also have this altered microbiota that allow it to be hard to be able to predict BV using the microbiome data? So our questions were, how do we accurately predict, can we accurately predict BV in its HIV positive data set using machine learning models? And then can we identify key bacteria that are population and disease specific? So from our asymptomatic data set to our symptomatic data set in our HIV positive data set. And previously in our asymptomatic and symptomatic work, we also narrowed this down to even looking at it by race and ethnicity. So this just basically gives an overview of the data sets that have been used in the past. So we used the Rebel data set that was 394 women with asymptomatic BV. And we had pH and Nugent scores for these women, as well as some demographic data. So they were composed of white, black, Asian, and Hispanic women. We had a Srinvasan data set, which was symptomatic bacterial vaginosis, and that included 211 women. This group had a deeper description of ethnicity. So we had black, white, Asian, Native Hawaiian, Pacific Islander, and mixed. And then this Tanzanian data set, which is our HIV positive data set or cohort, had 118 women, 50% of which were BV positive, and these were African women, so they were all black. And so the way that we first kind of went about this was looking at which models may perform well based on the variation and the distribution of the microbiome diversity, right? So we trained 4 models. We looked at the logistic regression, random forest, support vector machine, and multi-layer perceptron. And we evaluated each model for performance on each ethnic group in the symptomatic and asymptomatic data sets. And we basically looked at these specific metrics. We have balanced accuracy that I'm going to show today, false positive rate and false negative rate, just because these are important factors that contribute to accurate diagnosis and treatment for the disease. And so in our symptomatic data set, what we found was that generally there was a disparity in diagnosis, and this work was actually published in MPJ Digital Medicine in 2023, but we found that if you look at the total, it's pretty high accuracy for diagnosis for BV using the 16S sequencing data, but there were lower performance, especially for Black and Asian groups, yes, and Hispanic as well. And we did a deeper dive to understand maybe why this is in this particular paper. But in our asymptomatic group, we saw a kind of similar trend. And what we hypothesized is that it's the diversity of the microbiome. And the diversity of the microbiome that is healthy in some populations is flagged and allows them to have higher false positive rates when they're swabbed or when they're doing the Nugent scoring. So in our asymptomatic data set, we saw a similar trend here. We have it labeled a little differently. So on the bottom, we have the different classifiers that were used. And then on this axis, we have race. So we have white, black, and other. And what we found, again, was that it performed decently well across the board, but there were differences in performance when looking at the different ethnic groups. So, and this work was actually just recently published as well in MPJ Women's Health. We did a deeper dive on trying to understand why and extracting features there as well. So when we looked here, we also wanted to look and see if possibly training the classifiers, training the models using population-specific data improved performance or degraded performance. And what we saw was basically performance was best when we used the full data sets to train our models and then test the performance of the models. And so I think it goes back a bit to the the plenary talk that we had yesterday, where he talked about data and how we need actually like clean data, transparent data, data that represents lots of different groups, because then that will allow us to improve our accuracy, and our models are only as good as the data that we're inputting into them. So to transition then to this group of women who have are living with HIV, we also wanted to see, okay, so how are these models able to predict bacterial vaginosis in these women? And what we found is that all of the classifiers that we used kind of generally performed similarly across the board, maybe like a 0.75 for our balanced accuracy, precision, and recall a little bit lower. And then when we look at the false positive and false negative rates, they're about 0.2 here for all of them. It performs the different classifiers, they basically perform about the same across the board. But we wanted to also compare their performance to the other data sets. And what we found is that the models actually performed much worse for the women with HIV compared to those who did not have HIV when we looked at the asymptomatic and symptomatic data sets. So you can see here that the false positive rate is higher in the random forest model, logistic regression, SVM, and MLP. And then if we look at the false negative rates, we see kind of a similar trend here for this cohort versus the others. So we wanted to understand why, like why is this happening? And so we did a TISNI projection here to kind of understand and see how the bacteria was distributed, or it takes this multi-level data and kind of plots it in 2 dimensions. So what you can notice here is that if you look at the Tanzania data set, so the red is the red is BV positive, and then the blue is BV negative, right? So you can see here there's quite a bit of overlap in those diagnoses here, even on the TISNI projection. And as I said before, they're diagnosed by Nugent score. So a low Nugent score is healthy, whereas a higher Nugent score is BV positive, and you can see the intermediate score. So you can see here that it's kind of all intermixed together when trying to diagnose here with the Nugent scores. But when you look at the community state types, so community state types give a characterization of microbiome based on the dominant bacteria, right? So community state type 1 is Lactobacillus crispatus, which is known to be healthy. So we don't really have a lot of that in this data set. 3 is like kind of can go one way or the other. It can be either dominant in a unhealthy or healthy state. It's like kind of one of these transitory dominant bacteria, Einers. And so you see there's a lot of Einers there. And then community state 4 is like your very diverse microbiome that has a lot of anaerobic bacteria. And you would expect that your 4 would be BV positive. So we're able to kind of see that these cluster a lot better by community state type, the type of dominant bacteria, versus the BV diagnosis. Whereas if you look at the asymptomatic and the symptomatic data sets, they cluster a lot better with the negative and the positive diagnoses, even with the intermediate versus high and low. And then they also do cluster by the dominant bacterial characterization for the community. So we also ran feature selection, and so and did feature importance using permutation. And what we found is that there are different bacteria that are important for accurate prediction across the groups. And this is where we come and we're using this as kind of like hypothesis generating. So we see Gardnerella vaginalis is very important across all 3 groups. L. Einers was the most important feature for the Tanzania data set, which also tracks with what we saw with the TISNI plots. And really what this does is it gives us a very interesting list of bacteria that we can go then and study in our lab and start to think about how these things contribute to the etiology of BV. How can these things contribute to better therapeutic strategies that might be personalized or probiotic or, bacteria-based. And so, in conclusion, I'll say that for personalized medicine and diagnosis, several scenarios must be considered. A model may generally perform well, but in order to get, again, to the more personalized medicine, we have to understand the demographic information and how that allows us to delineate differentiation in performance between groups. Things can also be not even only ethnic, but environmental. So again, I think I heard another speaker talk about, will we use the same thing in Houston, that we would use in Florida? Will we use that same data? Is it the same? And so I believe that these are important things to consider and things that we're also trying to address in our lab through also our own data collection, because we're also collecting swabs and characterizing them and trying to improve those data sets. So that is all I have. I'd like to thank the team, to a PhD, one boy, postdoc, Deandra, Dr. Ojo, Cameron, and Dion, collaborators at UF and funding here as well. So thank you all.
Thank you very much.
Thank you for sharing your great work. I had a question regarding sampling the men. If these women were all sexually active, were they not sexually active? I saw a study earlier this year by the New England Journal of Medicine on how when they treated both the symptomatic female partner and the asymptomatic male partner with antibiotics, the rate of BV went down by half and there was no recurrence. So are these questions and factors considered when you're selecting data for these models?
Yeah, no, that's a great question, and I think really relevant to the field right now. One of the things that we are also doing with our current collection is also doing questionnaires, right? So we can get some of the metadata that would also allow us to understand what are some of the other driving factors. But I do definitely think partner interactions as well is definitely a key component that generally is not studied or not, the data is just not collected with the vaginal swabs.